just coming out of this.
Oh, do you want to turn your volume down then?
No, I can keep track of this.
Well, should we just tell people to ask in the chat then if it's being like, or like raise your hand and then like I'll stop and you can turn your volume on.
Yeah.
Okay.
All right. If you have questions and you're on Zoom and you want to ask them with your voice, then raise your hand and then we can hear you after that. But if you don't raise your hand, then like hear you.
Yeah. Cool. Okay. So are we all good to start? All right. So we're going to be going over the exam 3 material, which is going to be topics 6, 7, and 8. So we'll start with topic 6. And topic 6 was all about signal transduction. So first we'll go over just like the basic material. So we know that the signals are generally small molecules or proteins. And if the signals are polar or large, they're going to bind to cell surface receptors in order to affect change within the cell. So these cell surface receptors are proteins that are embedded in the membrane of the cell. And when they bind, the signals are binding to the cell surface receptors. They're not entering the cell. They're kind of causing like a domino effect reaction. So they're gonna, is everything fine?
Yeah, keep going.
Okay. So they're gonna affect the conformational change in the receptor once the signal binds to it. And this is just going to begin the reaction that is signal transduction. And we know that this occurs in 3 stages. So there's reception, which is basically signal binds to receptor. There's transduction.
Yeah, I just want to see if we get
Okay. Which is everything that goes on in between reception and the effect happening in the nucleus. And then we have the response, which is the effect that was caused by the signal. Yeah. Okay, cool. So there's 2 general outcomes of signal transduction. So signal transduction can cause an existing protein function to be altered, and this is going to be a lot faster compared to the other outcome, which is altered protein synthesis and gene expression. So this is a lot slower because we're causing activation and silencing of a gene. So for that one to occur, there's way more steps of like what's getting made, what's getting silenced versus just like, this is a protein we already made, like let's just change its function a little bit and send it up.
Okay,
it's on its way.
Like a question for class. In the second outcome, which is if there's protein synthesis or gene expression, like a modification of that, what do you need? What's the last step of that signaling pathway before you get into gene expression? What is the point of signaling pathway? The signaling pathway will either turn something on or off. What is the name of that? Think back to what controls gene expression. There you go. I heard a whisper transcription factor, and I was perfectly correct. So basically what will happen is at the end of that signal transduction pathway, it'll usually turn on a transcription factor. In most cases, it'll turn on if you want to express something, or it can turn off the transcription factor if you want to stop expressing something, or turn on an inhibitor if you want to stop expressing something. Does that make sense to everyone? Yeah. Does everyone get why option 1 is faster than option 2? Because in option 1, you're like changing proteins that already exist. In option 2, you have to wait for like all of transcription and all of translation to occur instead of just changing a protein that's already made.
Oops, I moved on. Okay. So now we're going to talk about.
We have a question in the chat. Do small non-polar signals enter the cell?
Yes.
Yes.
So in case you're wondering, like the reason why like a polar molecule would have a hard time entering the cell is because we know that like the cell membrane has non-polar parts. So basically it'll be very hard for a polar signal to pass through the polar layer of the bilayer. But if it's small and it's non-polar, it can just go in, I guess. Yeah. Okay. So important to know, cells can receive more than one signal at the same time. And the cells that are being received, or the signals that are being received by the cell, are depending on what receptors are being expressed in the cell. So this is kind of how you control like what signals the cell is going to respond to. Like if it's getting a signal for, say, like differentiate, but it's not expressing that receptor, it's not going to be able to enter, like to bind to the receptor and like affect the change within the cell that'll cause it to differentiate. And also when a cell stops receiving signals, it undergoes apoptosis. So the cell is continuously receiving signals to like just survive and do like the basic functions of a cell that needs to survive. And so when those signals stop, the cell will die.
And we'll go into more detail with the specifics.
Yeah, everything in this beginning one we'll go into more detail in a couple minutes, but this is like the foundations. So also the same signal can be received by different cell types, and it can have different effects on the cell depending on what the cell is. So if we use adrenaline, for example, we know adrenaline is part of our fight or flight reaction, and it's produced in our brain. So if adrenaline is received by a cell in the heart muscle, it's going to increase your heart rate. If it's received by a lung cell, it's going to expand the air passages of the lungs so you can get more breath in. And if it's received by like your retinal or your pupil cells, it's going to enlarge the pupil in your eye to let more light in. So essentially, it'll have a different effect on the specific type of cell that it's being received by. Does that make sense to everyone? And now to go over signal amplification. Signal amplification occurs because of diffusion rate limiting kinetics. So basically, reactions between molecules are limited by the amount of molecules present, because for reactions to be occurring between molecules that are just kind of floating around, they need to be bumping into each other for that reaction to occur. So if we have more of each component of these reactions, we're going to get more reactions because they're going to be able to bump into each other way more often. Yeah, that's basically the point of signal amplification. So it's like the signal will be like received by the receptor, and this will cause like multiple downstream reactions instead of just sending like one downstream reaction, if that makes sense. Is there a question in the chat?
How does the concentration of the downstream components increase? Usually you just activate like a greater number of downstream. So like if you're looking at this picture, the activated pKa, each one of those will go and activate 3 target proteins. So that's the amplification. You're increasing the concentration of the downstream target proteins. Instead of having each activated pKa only activate 1 downstream target protein, in which case you have no signal amplification. You just have the signal moving forward. Does that answer your question?
Also, if anybody here has a question, just raise your hand and let us know. Okay, and now crosstalk. For crosstalk, we'll go into it a little more in detail, but it's basically just the pathways and the proteins can talk to each other. So if you look at this, this diagram here, like wherever you're seeing horizontal arrows is basically crosstalk. So it would be like the product of one pathway can then activate or inhibit another pathway. It can cause just basically any change to occur in a different pathway. So that's how they can kind of like different pathways can regulate each other in the cell, and they work together to get the desired outcome, if that makes sense.
So I'm now going to go over like a detail about RTKs because you guys do need to know that in the context of there's like a survival signaling pathway that you guys need to know. There is an example pathway that's on the next slide that we use in a lot of this class, and we will get into that in a second. But just so you guys understand, because that pathway is based on an RTK pathway, I just want to go over what RTKs are and how they work. So this diagram right here is super complicated, and you don't need to know like the ins and outs and every detail. You just need to know generally what happens, which is basically some signaling molecule binds to the inactive RTKs, which are those blue things, each one of those blue things. Once signaling molecules have bound to those, two of them come together and they dimerize, which means they like interact to form from two individual parts into one part. And that's the dimerized RTK. Once the RTK is dimerized, it can phosphorylate the other RTK. So the one on the right will phosphorylate the one on the left, and the one on the left will phosphorylate the one on the right. They don't phosphorylate themselves, they phosphorylate each other, right? And if they phosphorylate each other, what that means is that they're adding phosphate groups to the other RTK. Does that make sense so far? Right? And that only happens once they dimerize, and dimerization only happens once the signaling molecule has bound to it. So once it is phosphorylated, now think about like what that means, right? You have this protein and now there's a different group, a phosphate group. It's PO4 minus is the general phosphate group, has now been added onto that. So the shape of that protein has now changed, right? Like the chemistry of that structure has now changed. So now other proteins that are involved in the signal transduction pathway have a different surface on which to dock, right? If you think of them as like spaceships or something like that in the context of the cell, they need something to like dock and like kind of latch onto. Before that phosphorylation happens, they can't latch on, but once that phosphorylation happens, the shape of that RTK has changed, the shape of that location has changed, so something can latch on, and once it's latched on, it can activate and then do whatever it does downstream, right? RTKs are used in a whole like classload of different pathways, and this is just the receptor part, right? So from here down, you can have a million different types of effects depending on the specific proteins that dock and what they do and things like that. Does that make sense? Any questions on RTKs?
Real quick, if you want to switch, you can use like
Yeah.
the cursor on my left and point so people on Zoom can hear it. And I can switch out.
I still want the mic. Okay. Does anyone on Zoom have questions? You good? Okay. So that is RTKs and how RTKs work. So now moving to like an example, the example pathway that you guys see a lot. And you guys will see this will definitely show up on your, this like pathway, this diagram will probably show up on your exam. I can almost guarantee it. I don't know what kind of questions that you'll be asked, but just kind of understand what happens is that this is when the pathway is off. Keep in mind that every part of the pathway is still there. It's just in the off position, each part of that pathway. So once it turns on, the signal comes in. You have the two receptors dimerizing, just like they did back here. They're dimerizing, they're like coming together. Once they've come together, they'll phosphorylate each other, and that phosphorylation, you see the PO4s right here, activates them. Once they're activated, then they can activate TK2 by phosphorylating it. Then they can activate, or sorry, they can activate TK1 by phosphorylating it. That will activate TK2 by phosphorylating it. That will activate TFA, which is transcription factor A, by phosphorylating it. And that will go and carry out its transcription factor activity, which in this case is it's acting as an activator of transcription. So it's binding the DNA, recruiting RNA polymerase, and increasing transcription of some specific gene. Right? Well, I don't know the specific gene here because this is just like kind of a bare bones example. But the reason we're using this example is that so you guys can understand how like the pathway kind of moves forward. It's like kind of like a stepwise thing where one thing activates the next thing, activates the next thing. Every single one of these activation points is a opportunity for crosstalk, which Athena just talked about, or signal amplification, which Athena also just talked about, right? So if TK2 activates 10 TFAs, that's amplification. If TK2 is activated also by a different receptor, that's crosstalk. Does that make sense? Any questions? No? Okay. Any questions on Zoom? Okay. All right, so kind of what we've been talking about for a little bit now, just to go over phosphorylation and dephosphorylation, because that's a big way of how signal pathways are controlled. Phosphorylation and dephosphorylation can be used to turn on or off different proteins that like make up different steps in the signaling pathway. Essentially, usually that phosphate comes from an ATP. This is ATP structure. You can see it's got 3 phosphate groups, 1, 2, and 3. That's why it's ATP, is adenosine triphosphate. You pull off one of those phosphates to make it ADP, adenosine diphosphate, and you take that phosphate group. A kinase will take that phosphate group and attach it to some target protein, right? Kinases add phosphates to proteins. Then you can remove that phosphate group if you so choose, if the cell decides that that needs to happen, with the help of a phosphatase, which is a protein that removes the phosphate group, and that just leaves as a free phosphate group. And now you're back to the dephosphorylated form. Keep in mind that what's happening at the end of the day when you're phosphorylating or dephosphorylating a protein is you're changing the chemistry of an R group of some amino acid on this protein, right? You're adding the phosphate group to like an arginine R group or a tyrosine R group or something like that, right? And you're changing both the chemistry and the physical structure of the protein at that location. Does that make sense? Yeah. Okay. Keep in mind though that phosphorylation doesn't necessarily mean on or off or anything specific. Phosphorylating something can turn it on. Phosphorylating something can also turn it off. It depends on the specific protein and what the effect that phosphorylation does, right? If you're phosphorylating an active site, that could make the active site more negative, which might be what you want. It might be less negative, which might not be what you want. It can either be an on or off change. I just said negative. I just want to make sure that everyone understands that the phosphate groups are negatively charged. So if you're adding a phosphate group, you're adding a negative charge to your protein. Okay. So moving on to the ways that signaling pathways can be controlled. We talked about dephosphorylating and phosphorylating proteins and like different steps of the signaling pathway. Phosphorylating might turn something on or turn something off, a specific step on or off. You can always also have feedback loops, proteolysis, there we go, or endocytosis. Feedback loops basically means that one step in the pathway, a late step in the pathway, either activates or inhibits an earlier step. So it kind of goes back, exactly what it sounds like. It's a feedback loop. It forms a loop, it goes back and it either turns off something earlier, which is a negative feedback loop, or turns something on earlier, which is a positive feedback loop. So in a positive feedback loop, the more that the signal gets to its endpoint, the more times that signal will be fired in the first place. In a negative feedback loop, the more that the signal gets to its endpoint, the less times that signal will be fired in the first place, right? So negative feedback loops kind of turn themselves off. Positive feedback loops turn themselves on. You can also have proteolysis, and this is mediated by ubiquitin. So E1, E2, and E3 are proteins that find some target protein that they have that might be involved in a pathway and basically cover them or add to them, kind of like we added a phosphate group earlier. They add a protein, a small protein called ubiquitin. If you add a whole bunch of this ubiquitin to some target protein, that target protein will be degraded, right? So if you're trying to turn off a signaling pathway and you know there's some protein that's involved in that signaling pathway, a way to turn off the pathway can just be to degrade that protein, get rid of that protein so you don't have any way to get from A to C because you've gotten rid of B. Does that make sense? Right. And then receptor-mediated endocytosis. Athena mentioned earlier, that one thing that you need for a cell signal to happen is you need a receptor, right? So if Athena's trying to say something to me, but I don't have, like, in some language, and I don't have a dictionary for that language, I can't understand what Athena's saying, right? So a way to control cell signaling pathways and essentially turn them off is by getting rid of the receptor in question, right? You're actively tossing out the dictionary that you need to understand what Athena's saying. The way this happens is the signaling molecule will bind to whatever receptor it binds to. And as soon as that happens, if the cell's like, I don't want this signaling pathway to happen, it'll pull that receptor with its signaling molecule into the cell, break down that signaling molecule. And a lot of times the receptor will go back to the outside of the cell, or it might stay inside the cell. It just depends. Basically, that receptor and the entire cells, and its signaling molecule that it's bound to, is pulled into the cell instead of carrying out activating some downstream effect. Does that make sense? Yeah. I chose not to put the endocytosis picture in here because I thought it was more confusing. But yeah.
Somebody asks, is the receptor degraded?
A lot of times, no. And it actually goes back to the cell, the surface of the cell. That does seem kind of counterintuitive, but if you think about what the cell's trying to do, is it's trying to stop the signal. So this enables the cell to kind of catch the signaling molecules that are present in the environment on that receptor and then degrade those signaling molecules as well. So by not degrading the receptor, it's not just ensuring that its own cell doesn't take in that signal, but it's also ensuring that there's less of that signal going around in the environment for other cells to pick up. A lot of times, as if it's like a negative signal, like a viral signal or something like that. Make sense? Make sense for everyone? Sorry. All right. So that was all topic 6. Does anyone have any questions generally about topic 6 thus far? No? Okay. All cell signaling. You guys are good? Awesome. The next thing, topic 7, is the cell cycle. So these are my cells, right? Or my slides? Yeah. So replication is very high stakes. It takes a lot of energy. It can be good. It's usually good because you're getting your cell to replicate. You're letting the cell proliferate. In a multicellular organism, you're like getting more cells, healing wounds, doing whatever. So replication is a good thing, but it takes a lot of energy and it can go wrong really easily. If it goes wrong, it's really bad because you can result in cancer, which is a cell just kind of replicating out of control. So in order to replicate, to like carry out this process, they have to replicate their DNA, all of their DNA. They have to replicate their organelles, right? Like their mitochondria, chloroplasts, whatever. And then also physically grow. The cell has to grow to about twice its size so it can physically divide. And then you can have 2 cells that are the same size as the original cell. And this whole process of replication is governed by the cell cycle. And they ensure that the cell is in a good state to carry out replication and that it's safe to do so. Make sense? Okay. So cell cycle is 4 phases. Quick breakdown of those phases. G1 is growth phase and preparation for S phase. S phase is synthesis, which is when replication of DNA happens. G2 is more growth and preparation for M phase. In M phase, the cell physically divides. The M phase is broken into prophase, metaphase, anaphase, and telophase, which used to be called mitosis, which you guys might have learned in like high school. We don't use that term anymore, not so much, because I don't actually remember why, but it's not really used as much anymore. People go with M phase. And at the end of mitosis, you have cytokinesis, and all of that makes up M phase. Then there's also a G0 phase. I'm just going to mention that here. You guys don't really need to go into super depth about what that is and how that works. But basically, if a cell is not actively replicating, which not all of your cells are actively replicating. A good example is your neurons in your brain. Those don't really replicate that much. Those sit in a different phase called G0, which is basically just inactive, right? They're not actively trying to replicate. And they can enter and exit G0 from G1 before the G1 checkpoint.
Sounds good? Excellent. Okay, so a little bit more depth into G1, S, and G2. G1 phase is also called gap phase 1. That's what it stands for. This is when the cell creates all the necessary proteins and checks that it's ready to synthesize new DNA, right? It has all of the machinery that it needs to synthesize new DNA during this S phase. And at the end of this G1 phase, you have what's called a restriction checkpoint, or the G1 checkpoint, which the cell, it says either commits to cell cycle. It's supposed to say either commits to cell cycle or enters G0 and like stops, chooses not to divide. But that restriction point is the point, it's called a restriction checkpoint because it's the point of no return. Once it passes that, it's committed to going through the rest of the cell cycle. It's committed to going through S, G2, and M. And if at any point it really, really can't, if it like breaks so badly that it can't finish S or finish M or finish G2, apoptosis. The cell will just be like, okay, it's my time to bow out, and it will carry a controlled cell death. So the restriction checkpoint is a big deal because it really needs to know that I have to get through the rest of this process or I'm going to die. The next phase is S phase or synthesis phase, which is the synthesis of new DNA. The cell replicates all of the DNA that it has. And during this, throughout the entire S phase, you have something called the S phase checkpoint. That's just kind of constantly checking to make sure that as it's replicating the DNA, is there any DNA damage that is sustained? So the S phase checkpoint like kind of comes in as soon as DNA damage occurs, and it like fixes that damage before the cell moves on. Then you have G2 phase or gap phase 2, which is when the cell checks that the DNA was replicated properly during S phase and that it's prepared to physically divide in M phase. This is just a kind of more growth, more prep for M phase. And then this has the G2 or M checkpoint at the end of G2, where the cell physically commits to physical division. Like kind of how at the end of G1, it committed to the division process as a whole, starting with DNA replication. Now it's committing to physical division. Any questions? There's really no point to this mic, is there? I just like pulling it. My arms are getting tired. I'm gonna turn this off. All right, M phase. Just a quick breakdown. Mitotic spindles are made up of something called microtubules, which are part of the cytoskeleton. Microtubules are just like kind of aggregates of protein that kind of more form what's in essence a dry spaghetti noodle. They're like rigid. They like can bend, but they don't like form angles or anything like that. And you have these dry spaghetti noodles throughout the entire cell, and this is what like organelles are, unless they're free floating, or like anchored onto these spaghetti noodles, or things to like walk along these spaghetti noodles to move things through the cell. The microtubules can connect to each other or to chromosomes by connecting to a protein structure called a kinetochore. The kinetochore is at the middle of a duplicated chromosome. You know how duplicated chromosomes kind of look like an X shape, right? Right at the point of that X, right in the middle of that X, that cross, is the centromere. And nope, centromere, centrosome, something like that. Centromere, right? Centromere, and in that centromere is made up of a kinetochore, which is that's just like a bunch of proteins that the microtubule can anchor to. Sounds good? Okay. So during M phase, this is when the assembly of these spindles occurs, these mitotic spindles from the ends of the cell to each of the kinetochores in each of the chromosomes, the duplicated chromosomes. And the chromosomes actually also condense, which is what, once they've condensed, they form these like little cross or X shapes, and they've attached to the mitotic spindles. They then segregate, which just means that the daughter chromosomes are pulled apart to opposite sides of the cell. I'm sure you guys have all seen like videos of this happening, or like diagrams, pictures, things like that. It's just the X's come apart into single strands, kind of. And then you just have all of your daughter chromosomes on each side of the cell. During the M phase, you have one more checkpoint. It's called spindle assembly checkpoint. It happens right before segregation. Once all of the spindle assembly has happened, and it makes sure that all of the spindles have formed correctly and all of the that each chromosome is attached to two microtubules, one coming from each side of the cell. Right, and it won't proceed until that has happened. Does that make sense? Everyone's good? Okay. Any questions on Zoom? All right, so a quick recap of your checkpoints. You have 4 checkpoints. The G1 checkpoint, which happens right here at the end of G1 before it can go into S. That's a restriction checkpoint. It checks to see if division is viable. So if it is the right size, if it has enough nutrients, if there are any extracellular signals telling it not to replicate, and that the DNA that it originally has is undamaged and totally okay, this is the point of no return. Then throughout the entirety of the S phase, you have the S phase checkpoint. This is where it's checking as DNA replication is happening. It's making sure that that process of DNA replication is happening properly, that there's no replication errors or damage to the new DNA or the old DNA. Excuse me. And if there is any kind of damage or any issues with the replication, the cell can pause replication at that moment, fix the damage or fix the errors, and then continue in replication. You then have the G2 checkpoint, which is the post-replication checkpoint. It's after all the DNA replication has happened. It's still checking to make sure that the DNA replication happened right. And if there are any errors, again, it'll fix those errors before it moves on. It also just makes sure that all the DNA was replicated, because the S phase checkpoint is just checking that the process of replication is happening right, but it won't check to see if there's like any DNA that wasn't replicated. G2 checks to make sure that all the DNA that was supposed to be replicated has replicated. And then during M, in here, you have the spindle assembly checkpoint, which checks to see that each chromosome is attached to 2 kinetic core microtubules and will not proceed until each chromosome has been attached to 2 kinetic core microtubules. Make sense? Okay. This right here, not this exactly, but something to this effect.
There's a question that says, is it also part of the M phase?
Is what also part of the M phase?
The SAC.
Right. The SAC happens during M phase. Yeah, so SAC is occurring during M phase. Make sense? Okay. I would have something like this diagram, like a much far more simplified version of this diagram on your cheat sheets, just so you know like what the general idea of, and like in that diagram, I would like label like what does G1 do? What is the G1 checkpoint looking for? G2, what is the G2 checkpoint looking for? S phase, M phase, etc. Just like have something to this effect on your cheat sheets for the overall cell cycle stuff. Okay. Everyone's good on the cell cycle and how it works and what it is and what happens when? Yeah, excellent. Now, how do you regulate the cell cycle? How do you actually stop things at the checkpoints or push things through the checkpoints? An important part of that they're called cyclins. They're like the smaller proteins, and they're really important regulators of the cell cycle. And they bind to something called cyclin-dependent kinases, or CDKs. And when a cyclin binds to a CDK, it helps to activate that CDK. And CDKs are really what does the stuff, but the cyclins are used to regulate those CDKs. They're used to turn CDKs on or off. So they changed. You can see right here in this graph, the amount of cyclin will vary over the course of the cell cycle, depending on what stage you're in, right? So if it's G1 or S cyclin, G1/S cyclin, that's a specific type of cyclin that's necessary to go between G1 and S. G1 cyclin is just kind of present throughout the entire thing. S phase cyclin is made during S phase. G2 cyclin is made, or sorry, not G2 cyclin, M cyclin is made so that it will be at its peak to push into M phase, right?
Yeah.
And you can see that the presence of cyclin kind of correlates with the stage in the cell cycle, because when there is cyclin present, they will activate the corresponding CDKs, which will activate whatever it needs to activate to push the cell through the cell cycle at that time. So they're made and degraded based on what they need to like be pushing, right? Whether or not they need to be bound to CDKs. So G1 cyclins will be synthesized and degraded differently than S cyclins, which will be synthesized and degraded differently than M cyclins. Make sense? Excellent. And the CDKs themselves, they're also kinases, which means that they phosphorylate things. They promote the cell cycle. They're really controlled by three factors, right? Whether they're bound to a cyclin, whether they've been phosphorylated by CDK activating kinase or CAK. This is necessary. The cyclin binding is also necessary. And the inhibition is a process where WEE1 phosphorylates the CDK. So what happens is you'll have a CDK, it'll bind the cyclin, it'll get phosphorylated twice, one by the CAK and once by the WEE1. In that stage, the cyclin is actually still inactive. That WEE1 phosphate needs to be pulled off. It needs to be dephosphorylated to be for the cyclin to be fully, for the CDK to be fully active. Does that make sense? So you need the cyclin to bind, you need phosphorylation from CAK, and you need dephosphorylation of the phosphate that WEE1 has put onto it. Make sense? Yeah, okay. Then there are other CDK inhibitors as well. Those are specific to whatever is happening in the cell at the time. They will go and activate some kind of inhibition on the CDK. We're not going to go into any specifics. You don't need to know any specific examples, but just know that it is possible to do that, especially if there's some kind of cell stress, like some environmental stress or DNA damage. The CDKs will be actively inhibited until that stress is resolved. A lot of the time, these inhibitors are actually normal and a normal part of the cell cycle and its regulation. And then another way of regulating CDK activity is by regulating the concentration of CDKs themselves, similarly to how you regulated cyclins. You can also synthesize or degrade CDKs depending on when they're necessary and how much of them are necessary. Make sense? Yeah. Is there a question?
Yeah. Are the other inhibitors proteins?
Yes. For the most part, yes. I can't think of an instance where they wouldn't be. It's like kind of stuff, it's kind of like WEE1 is technically an inhibitor of a CDK, but they're CDK specific ones. So like when it's in S phase, there might be a specific inhibitor that comes in and blocks the active site or something like that. All right. One more big part of cell cycle regulation is P53. P53 is a transcription factor that is a central regulator of cell stress responses and is often involved in various checkpoints. When DNA damage occurs, which can generally occur during S phase or can be caught in G2 phase, P53 gets phosphorylated and is activated, right? Once it's activated, P53 acts as a transcription factor for a protein called P21. And P21, just like we were just talking about, is a CDK inhibitor. So it'll inhibit G1 cyclin CDK complexes. And once P21 has inhibited that, it won't be able to push forward in the cell cycle, and you'll get some like an arrest of the cell cycle. So it won't go past the checkpoint. P53 is also involved in signaling for apoptosis. So if it is carrying out the transcription of P21, and that's not, and nothing is happening to that damaged DNA, if that damaged DNA is not getting fixed, despite the fact that we have stopped in the cell cycle and we aren't going forward, eventually P53 will pull the trigger and be like, okay, this has gone on for long enough. Obviously, whatever problem that we're having is not going to get fixed. So apoptosis happens. It signals for apoptosis and the cell dies. Make sense? Okay. also want to make sure that I'm leaving enough time for you guys to like write notes. Are you guys getting enough time for all that? Yeah. Question in chat? Excellent. All right, so now moving to cancer genetics. If you screw up something in the cell cycle, a lot of time you can get the cell replicating out of control. There's really two things that can happen is that one, the cell just won't replicate, and two, the cell will replicate too much. And you'll go through, kind of spin out through the cell cycle out of control, and that is cancer. You're getting uncontrolled replication. So keep in mind that the cells are constantly receiving signals, like in the signaling pathways that we talked about. Athena mentioned a survival signaling pathway, and we talked about the RTKs. There's an RTK pathway that is a survival signaling pathway that is constantly telling the cell not to carry out apoptosis, right? That you're okay, cell, you're doing great, your DNA is fine, don't commit controlled cell death. And the cell kind of exists in this balance between proliferation and survival cues versus death and not growth cues, growth inhibition cues. It's in this like state of homeostasis where it's kind of balancing between all of these and hopefully surviving and hopefully proliferating correctly as needed. And depending on which pathways are the strongest, the cell will carry out whatever final function, whether it's replication or survival or death or proliferation or whatever. So if you have a gain of function in the survival and proliferation cues, then or a loss of function in death or inhibition cues, you can result in cancer because the cell is not dying when it's supposed to, and it's replicating when it's not supposed to. Sounds good? That's a genetic basis for cancer. Okay. So there are two types of genes that are really involved in controlling this process and ensuring that the cell doesn't kind of spin out through the cell cycle and result in cancer. One of those is proto-oncogenes. Proto-oncogenes are genes that are normally used for the growth of cells. If you think about the cell as a car, then this is the gas pedal, right? You want to be pressing the gas pedal. You want to be pressing the gas pedal to go, but you want to be pressing in a controlled fashion, right? You don't want to go over 40 miles an hour in a school zone. If you really lay on the gas, right? If you have a mutation that causes a gain of function in the proto-oncogenes, which means that the proto-oncogenes do their job, their end resulting job, better than usual. Gain of function, they're doing it better. They become an oncogene. This is your floor in the gas in a school zone. 60-40 chance you're gonna cream a kid. I don't know why I wanted that metaphor. That was really dark. That's fucked up. I'm sorry, guys.
You really gotta know when you
Yeah.
won't forget it.
Yeah, exactly. Cancer is not good. Neither is hitting kids. I don't know where to go from here. Okay, and then you also have tumor suppressors, which acts as the brakes in your car, right? They're making sure that you don't go over 40 miles an hour. And if you do go over 40 miles an hour, you can brake and slow down as needed, right? They regulate proto-oncogenes and make sure that the proto-oncogenes don't become oncogenes. They will stop the cell at a checkpoint if needed. What is one that we just talked about, Athena? Athena wasn't paying attention. P53. P53 is an example of a tumor suppressor. Okay. It's supposed to be like 20 or something like that. I passed my driving test. Yeah, it's fun. Yeah, so P53 is a tumor transcription factor for P21, which is a CDK inhibitor and stops the passage into the S phase, which means that it is stopping, like P53 is acting as a tumor suppressor and stopping the cell from moving forward. If you break your tumor suppressor, if your brakes don't work, your brake lines have been cut, your car is going to move out of control. Even if your car is still moving at 40 miles an hour, right? Even if your proto-oncogenes are just proto-oncogenes and not oncogenes, if there's a wall coming up, you still can't brake, right? You're still going to hit that wall or that kid at 40 miles an hour instead of at 80 miles an hour. So it's not ideal, right? It's not going to go as fast through the cell cycle, but it's still going to move through the cell cycle in an uncontrolled fashion. Do you get what I'm saying? So you can still get cancer just by breaking a tumor suppressor, you can still get cancer by breaking or having a gain of function in a proto-oncogene that makes it an oncogene. A lot of times the tumor suppressors will check the oncogenes and ensure that they don't go out of control. In reality, none of this is actually going to result in cancer because you need a lot of these mutations to happen over time, right? Breaking one proto-oncogene or turning one proto-oncogene into an oncogene or breaking one tumor suppressor is not going to get cancer. But over time, your cells, my cells, everyone's cells are mutating as they divide. A lot of my cells will pick up individual mutation. My P53 will probably break, but the cell will realize that and carry out apoptosis. But let's say that P53 break slips through, then you get an E2F break that slips through, you get an RB break that slips through, and eventually the cell that has built up so many oncogenes and has broken so many tumor suppressors that now it can spin through the cell cycle and you can have a cancer phenotype, right? So in reality, cancer is to build up a whole lot of these mutations. No one mutation is going to do it, but one mutation might break the camel's back. Make sense? Okay.
Can too much apoptosis cause cancer?
No. Yeah, because apoptosis is controlled cell death, right? Cancer is when you're having a cell replicating out of control. So if you have apoptosis, then the cells are dying instead of replicating out of control. So instead of too many cells, you're having too few cells. Uncontrolled apoptosis, like crazy amounts of apoptosis, will still be bad. It just won't result in cancer. It's actually called necrosis, where like big parts of your body will like die. That happens a lot to like people get them in like kidneys and livers and stuff like that. And it's not ideal. Yeah. That actually is what happens, I think, when you get frostbite. Yeah, that's why your fingers turn black and they fall off.
Or when people get diabetes, then
Yep.
they start losing their toes. Yeah.
Exactly. Diabetes and frostbite. Yeah. This is a really dark review session. Okay. Did that answer the question? So moving forward, I actually just mentioned E2F and RB. These are two other proto-oncogenes / tumor suppressors that you guys should know. E2Fs are transcription factors, kind of like P53. It's just that they're transcription factors for things that move forward in the cell cycle instead of things that stop the cell cycle. So E2F, like P53, is a transcription factor, but it has the opposite effect. They're transcription factors for the cyclins we were just talking about. So they increase the presence of certain cyclins, depending on where you are in the cell cycle. They'll increase CDK presence, they'll increase various checkpoint regulators, DNA replication machinery, DNA repair machinery, all that stuff that's necessary to push the cell forward and have it replicate. If E2F is overactive, it can become an oncogene instead of a proto-oncogene, and the cell can kind of slide by a checkpoint when it shouldn't. Okay. Then you have RB, which is the full form is retinoblastoma protein. You'll never see it called that on your exam or anything like that. It's just going to be RB. RB is a tumor suppressor that actually controls E2F. So it stops, it binds to and represses E2F. So in its normal state, actually, RB is bound to E2F because you don't want the cell to be making all of the cell repair and replication machinery normally. So the normal state is RB is bound to E2F. Actually, what happens is RB gets I think it's phosphorylated, right?
What is?
RB gets phosphorylated?
Do you have a picture of the thing on you?
I don't think so.
Okay, you can pull that up at the end.
Yeah.
I would put details from that on the review side of what gets phosphorylated by what. You know the one.
On
that's
the
all
like the
over
cheat sheet? What?
On the cheat sheet.
Yeah,
It's
I
like-
don't know what you're talking about. You know
I'll
what you're
pull it
talking
up.
about, so you can pull it up.
I'll-
What happens is RB is like bound to E2F normally and keeps E2F from interacting with DNA, like sequesters it. Eventually RB, when it's supposed to, when you're getting the cell moving through the cell cycle, RB will get phosphorylated and it'll change the shape of the RB. So it comes off, it can no longer bind to the E2F. So E2F is now free to go and transcribe the DNA and do whatever it does. Right? So RB is a tumor suppressor, just like P53, and E2F is a proto-oncogene. One more thing I want to go over is you mentioned RTKs earlier, and your like your unspecific example pathway that's based on RTKs. RTKs, there's an RTK map kinase pathway that's responsible for survival signaling, right? So like I said earlier, if the cell gets too much of a signal to survive, it'll survive out of control, which means that it can replicate out of control. It'll survive when it's not supposed to survive, even if there is huge problems with the cell or the cell's machinery or the cell's DNA. So a gain of function in the signaling pathway, if this signaling pathway is overactive at any point along that pathway, you can get an oncogene, right? You can get the cell surviving out of control. The opposite of a survival pathway would be an apoptotic pathway, but we've talked about that to some degree now.
Could you go back to RB what it is?
Yes. So RB binds to E2F and stops E2F from carrying out its activity. Yeah, that's the gist of it. It's that when E2F is supposed to carry out its activity, RB gets phosphorylated and it's pulled off of the E2F. So then the E2F is now free to interact with the DNA and act as a transcription factor. Does that answer your question? All right, you're up.
I'm just gonna exit out.
Well, I can just go out and like it should be. Okay, so this is what I was just talking about of like what I personally find the most helpful for remembering all the details of like what activates what and like what inhibits what and just basically all the steps of like getting the cell to commit to the cell cycle. So like me personally, like I would write this down, like the information from this down, just because in my opinion, this helps me the most. This is just from her slides. I think it's from the third video for week 7. Okay. So now to go into topic 8, which is enzymes. So what are enzymes? Enzymes are proteins. And we know that cells need chemical reactions to occur for like every single function. And they need to occur quickly and they need to happen often. And enzymes are these proteins that can create and break covalent bonds, but creating and breaking covalent bonds requires energy. The other interactions you can see in the cell, like hydrogen bonds or like van der Waal interactions and whatnot, those don't require like an addition of heat or energy because they're kind of occurring due to like the transient nature of like electrons. They can move around and whatnot, whereas the creation of covalent bonds is a process that requires an input of heat or an input of energy. Enzymes help in this process by catalyzing or speeding up these chemical reactions, but the enzyme is not changed by the reaction that occurs. And the enzyme allows these reactions to occur in mild conditions. So if you have an enzyme, then you're not going to have to heat up the cell to make this covalent bond or break this covalent bond. You can do it without having to really change the conditions the cell is in. So how do enzymes work? Essentially, they lower the activation energy of a reaction. And they do this by stabilizing the transition state. The transition state is like the in-between product of the reactants and the products. And this is like a really unstable state. So it has a lot of free energy. And by stabilizing this, or the enzyme is going to lower the amount of activation energy that's needed for the reaction to occur. However, enzymes do not change the free energy of the reactants or the products. So there's still the same amount of energy in the reactions and there's reactants and still the same amount of energy in the products. It's only changing the energy of the transition state. So the total free energy of the reaction is not going to be changed by the enzyme. Does that make sense to everyone?
There was a question process as opposed to signal pathway stuff. The question is, does the pathway have a gain of function signal is received more times than usual? I don't really understand what that means. I took that because there's a lot of single molecules that get picked up by the receptors frequently.
Well, I think like gain of
Gain of
function
requires a mutation.
is what?
It requires a mutation.
Yeah, so I think it wouldn't be like the signal being received constantly. It would be like the receptor has a gain of function mutation such that like it's not going to release the signal and it's going to like keep activating the pathway, I feel like, right? Yeah, so if it's receiving the signal a bunch, I don't know if you could call it like a gain of function of the signal, but like a better example would be like gain of function of the receptor. It's not going to let go and it's going to keep on activating.
Yeah, and so like gain of function of the receptor would be like it binds signal instead of binding one signal and carrying like activating one downstream effect. It binds signal and activates, just keeps activating the downstream effect and never stops.
Yeah.
That would be-
Yeah, okay. Does anybody have any questions about this? Yes, yeah. Anyone else? Is the chat good? Cool. So now we'll just go over like the parts of an enzyme reaction just to be like comfortable with the terminology. So you have your substrate and that's what's going to bind to the active site of the enzyme. And this is like what the enzyme is causing change to. You have the product or products. This is what's formed by the reaction. I put two examples for the pictures. You can have like two substrates come in and the enzyme is going to form a covalent bond between them and they're going to stick together. Or you could have one substrate come in and the enzyme is going to break the covalent bonds between them and they're going to separate. So then you'd have two products. And then you have your enzyme substrate complex, which is just when like the substrate has binded to the active site and they're sitting together. So it's just the physical association of the two. And now for enzyme action. So the substrate binds to the active site of the enzyme through non-covalent bonds. And substrates have a much higher affinity for the binding site than the products do. And this is like necessary for the cell so that once the product has been made by the enzyme, it can be released. And the enzyme can take in a new substrate and do it all over again. So if the product had a high affinity for the binding site, it wouldn't want to leave. And it would be hard for the enzyme to keep on doing its job, basically. And now to go over product formation. So the two things that affect product formation are substrate concentration and enzyme concentration. However, the enzyme concentration is usually pretty constant. It's not going to go up and down a lot, whereas the substrate concentration can change. So if we look at the graph right here, we see that eventually our graph or our rate of product formation is plateauing. So it's not really increasing anymore. And this happens because the enzyme can become saturated. It can become saturated by the amount of substrate that's in the area, because adding more substrate is not going to increase the rate of product formation if every enzyme's already working, basically. The enzymes aren't looking for an substrate anymore. It already has one at its active site. So yeah, basically it's just has turned everything there into product, or that it's limited by the amount of enzyme present. So adding more substrate isn't going to change anything if there's a limited amount of enzyme. Does that make sense to everyone? Is the chat good? Okay, yeah. And this like little silly picture down here is nice. Because you can see like all their active sites are taken. So the fact that there's more substrate present doesn't make it go faster. Oh. See,
All right, yes.
Yes, but if generally the amount of enzyme does just stay constant, it would be the amount of substrate that changes. But if you were hypothetically to add more enzyme, you would have more enzyme there to take up substrate.
Yep. Yeah. What you're talking about gets into some like more advanced stuff that you can do with Michaelis-Menten kinetics that we don't go over in this class. You're a biology major?
Yeah. Kind of.
Okay. If you take like upper level biochem classes, like 420, 423, 424, they go more into this. And essentially what happens is the math that you do accounts for enzyme concentration. So you essentially divide by unit of enzyme. So like the enzyme concentration doesn't actually matter for when it comes down to math and stuff like that. But the stuff that you get from the Michaelis-Menten kinetics, which is the binding affinity and maximum catalytic rate, is not dependent on the enzyme concentration, right? So binding affinity, what you can derive from Michaelis-Menten graph is for like one given type of enzyme to one given type of substrate, not a concentration of enzyme to a concentration of substrate. Do you get what I'm saying?
Yeah.
Okay. Michaelis-Menten kinetics is a way of modeling enzyme kinetics. Kinetics means reaction rates and speeds. And this is kind of what we've been talking about, but I'm now going to talk about more like what you can actually derive from Michaelis-Menten graphs and what's the whole point of that. Michaelis-Menten kinetics is used to determine a specific enzyme's binding affinity to a substrate and maximum catalytic rate or reaction speed for a substrate. And if you Google Michaelis-Menten kinetics, the biggest thing that you can see is the Michaelis-Menten equation. And we don't go over that in this class. That's like one of the things I was talking about. We don't go over that in this class. You don't need to know that for this class. You might see it in like Gen Chem. Yeah. Reaction rates. So to study the enzyme reaction rates, we plot the initial rates of an enzyme against various substrate concentrations to get the graph of initial velocity over substrate. And eventually what happens is the substrate becomes saturated and all of the active sites on the enzymes in the solution have substrate attached to them, right? If you look at this graph right here, here you have 5 units of enzyme, right? But there's only 1 unit of substrate. So only one of the 5 units of enzyme is being taken up. Once you get up to the top, no matter how much enzyme or substrate you add, you've taken up all of the possible active sites, right? So that's why as you increase in substrate concentration, the velocity stays the same, is because the limiting factor there is not the amount of substrate and the amount of open active sites that you have, but the limiting factor up there is just the turnover rate. So how fast is this enzyme able to actually get one substrate, turn into a product, and get another substrate, right? Because all of the active sites are being taken up right here. So that's called saturation. The enzyme is saturated, so all the active sites are full. And this leveling point is Vmax, right? The initial velocity at that leveling point is the Vmax. And that's the maximum possible initial velocity that you can get for an enzyme or substrate. That's a very, very important parameter of enzymes and their reaction to the substrate. Another important parameter is Km, which is the concentration of the substrate when the initial velocity is 1/2 of the Vmax. So Km basically tells you how fast you'll get to Vmax. And because it tells you how fast you get to Vmax, it's inversely proportional to the affinity of the enzyme to the substrate. So if it takes very little substrate to get to 1/2 of Vmax, that means that the enzyme is picking up that small amount of substrate like that, right? If it takes a whole lot of substrate to get to 1/2 of Vmax, that means that despite that you have a whole bunch of substrate around, the enzyme is not picking it up as much, so you're not filling half of those active sites as easily. Make sense? So there's an inverse relationship there. Yeah. Does that make sense for the Michaelis-Menten stuff? Does everyone understand this and how to read one of these graphs? Yeah. Any questions in the chat? Okay. Then we're going to get into the stuff that can interact with enzymes and change enzymes. First thing is cofactors and coenzymes. They're small molecules that are necessary for a specific function of certain enzymes. Cofactors are inorganic. They're usually metals that are required for the protein or the enzymes to function. A lot of times it's zinc, iron, or copper. You can also have like small inorganic compounds. The other option is a coenzyme, which is an organic molecule, which means that it's carbon-based, often in the form of vitamins. Actually, didn't know that are needed for the enzyme to carry out its function. Right, so vitamins are a good example of a coenzyme. Actually, that's true. Vitamins, I just remembered from Biochem 423. We talk about how vitamin C is a big example of a coenzyme. That makes sense? So you have something called an apoenzyme, which is the enzyme without the cofactor or coenzyme, and then you have the coenzyme or cofactor come in and it becomes a holoenzyme. And once it's a holoenzyme, it can actually interact with the substrate and carry out whatever activity it needs to carry out. Sounds good. I've got a confused face and everyone else is writing. Yeah, you guys good? Okay, any questions on Zoom? Nice. The 3 kinds of reversible enzyme inhibition: competitive, non-competitive, uncompetitive. This is a huge pain in the ass. So I would write this on your cheat sheet. The difference, especially between non-competitive and uncompetitive, because that's basically the same name and it means 2 very, very different things. So I would write down what each thing does and how that changes the Vmax or Km, which I'm going to go into in a second. These are reversible enzyme inhibition or inhibitors, right? Modes of inhibition. Because they're reversible, it means that they can be removed and their inhibitory effect can be reversed. You're not carrying out a permanent post-translational modification to the enzyme, right? The actual protein that is the enzyme is staying the same. You're just adding an inhibitor that has some sort of inhibitory effect. And this inhibitor can come off if you need it to. So competitive inhibitors bind to the active site of the enzyme and block the substrate. They physically block the substrate from binding, kind of like how the inhibitors block RNA polymerase when it comes to transcription. It physically blocks the substrate from binding to the active site, so the inhibitors bind to the active site. The effect on the Michaelis-Menten equation and Michaelis-Menten graph, is that you have an increase in the Km of the enzyme substrate pair, but not the Vmax. The Vmax doesn't change. The rate at which the enzyme carries out its catalytic activity doesn't change. Remember how that's the limiting factor once you hit, what's the word I'm looking for? Saturation. There you go. Once you hit saturation, the limiting factor, the thing that controls speed is really the turnover. How fast does the enzyme actually carry out its activity? Here, you're not changing the ability for the enzyme to carry out its activity, but the ability for it to pick up the substrate, right? So if you think about what does Km correlate to, it's how fast and how strongly can the enzyme pick up the substrate. So the effect of a competitive inhibitor that is binding the active site and making it harder to pick up the substrate is that your Km increases. Yeah, remember that increase in Km is correlated with a decrease in affinity. Then you have non-competitive inhibitors, which bind to an allosteric site, which means a site that is not the active site, just some other site specific for that inhibitor on the enzyme. The binding of a non-competitive inhibitor to an allosteric site causes a conformational shift in the enzyme. That means some kind of physical shift in the enzyme that changes its catalytic activity and essentially turns off the catalytic activity. So this is what I was just talking about, is this does that. This does turn off the catalytic activity. It doesn't change the way the substrate binds as much, but it turns off the enzyme's ability to carry out its enzymatic activity. So the maximum speed, the Vmax, decreases, but the Km does not change. It stays the same. I was going to say changes the same. That doesn't make any sense. The Km stays the same, but the Vmax decreases because the catalytic activity has changed. Make sense? Perfect. And then uncompetitive inhibitors. These bind to the enzyme and substrate complex. Once the substrate has already bound to the enzyme, and keep the substrate from dissociating, right? So it doesn't really, it does change the catalytic activity, but it also decreases the ability for that substrate once it's, the way that it decreases the catalytic activity is that it decreases the ability for the substrate to leave once the enzyme has carried out whatever activity on it. So it decreases the Km because you're increasing the affinity for the substrate by just not letting it leave the enzyme. And it's also decreasing the Vmax because you're reducing the catalytic activity of the enzyme. Does that make sense?
You said it decreases Km, right?
Yes.
Okay, I thought you said it increases, but decreases. I don't know if
Oh, no, no, no.
I just want to make sure.
Yeah. It decreases the Km because it's increasing the affinity because it's not letting the substrate leave, right? And it decreases the Vmax because you're reducing the catalytic ability. So just to recap. What the hell just happened? Okay, yeah, that was last night. Okay, there we go. Competitive means an increase in the Km, no change to the Vmax. Non-competitive means a decrease in the Vmax, no change to the Km. Uncompetitive is a decrease to the Km and a decrease in the Vmax, right? It's important to recognize that this is a decrease in Km instead of an increase in Km because competitive has an increase in Km, but uncompetitive has a decrease in Km. Everyone's good? Yeah.
Yeah, no additional questions.
That was all of our slides.
Yeah, we have a practice Kahoot. It's pretty short, it's like 15 questions.
Kahoot.
Yeah, just to like nail home some of like the most important concepts and like things that can get mixed up.
Take it
Run
away.
through
Kahoot.
Slide.
And if you guys have questions about anything that we've talked about or any questions, feel free to ask them.
Where'd it go? Oh no, can you see the answers? Don't look, guys. How do I not share for a second?
Go back.
I know. No, I got stressed out because now everybody can see the
Hit
answers.
stop share first.
Stop share this.
Yeah. I'm gonna unplug this for a moment so nobody
Okay.
no one can see.
Stressed me out. No, wait. Sorry.
What the hell?
It's this.
Yeah.
And then I go up here. Go away.
Wait, yeah, save it
Save.
and then hit done. Or you
Oh,
can just
play
hit
now.
play now. Yeah.
Oh god. Play. Start. And then we'll plug this back in.
Now you can reshare.
Go to the Zoom.
Yep.
And I'll share my screen again. Yes. The music works.
Yeah, reshare it with the audio.
I think it is on. Everybody in the chat. Oh, someone's asking this. Just scared me. Will the slides be posted? We can send out a link to the slides. This also whole session will be posted on YouTube. And then somebody said, do you want to answer the differences between catalytic rate and turnover rate? And then everybody on the chat or on the Zoom, just let me know if you're having like, if it's okay for you to join from
Can
here.
you hear the music?
Sad. I don't know how
Damn.
to fix that. You probably find it on like Spotify if you want full experience.
No, it's right there.
Oh, I don't know how to do anything.
Just put it back.
That not work.
Okay, while Tina is doing this, rate, catalytic rate is from start to finish. If you put in the substrate, what is the rate at which you get product? right?
I'm gonna have to restart my laptop if we do that, I think.
Oh, okay. So let's not do that.
Oh, no, it's not working at all. Oh, it's muted. Right?
Nope, it's just not working at all.
Sad.
Okay, I'm just gonna assume everyone can hear us. So, the catalytic rate is like the rate that you get product from substrate. Like if you put enzyme and a product together, how fast do you get, or enzyme and substrate together, how fast do you get product? But the turnover rate is when it comes down to an individual like molecule of enzyme, an individual protein, and an individual molecule of substrate. Once that substrate has been bound to that individual molecule of enzyme, how fast does that individual enzyme turn that individual substrate into a product? Right. So it's less. Think of catalytic rate as like overall in the context of all of the product that you're adding and all the enzyme that you have, or all the substrate that you're adding and all the enzyme that you have, and turnover rate as for an individual molecule of enzyme and an individual molecule of product.
Okay. Is everybody good to start the Kahoot? Yeah, bummer. Okay, let me know if anybody's still trying to join.
Oh, no.
Can you do that? Okay, cool. Can you mute us though? Can you not do that and mute us at the same time? Like so my voice isn't echoing. Okay. I'm gonna start it now. You can't play it and have us muted at the same time.
I'm just gonna play Kahoot music for 10 hours.
Okay. Okay, good job, guys. It's like freaking me out to hear my own voice like that.
Yeah, I'm muting you.
Okay, that's like kind of loud. Turn it down a little. Okay, yeah, everybody got it right, so I don't think we need to go over that. If anybody has any questions, though, let me know. We can't stop them, even if you answered it correctly but are unsure. Nice. Okay, cool. So we'll go over this one. So if we go back here, we see that in step one, right, we have one signal binding to one receptor, and this receptor is activating multiple cyclic AMPs, right? And that all is happening from just one single signal and receptor interactions. However, step two we're seeing is between cyclic AMP and the activated PKA, right? But each cyclic AMP is only activating one PKA. So therefore, we're not seeing any signal amplification because each camp is only activating one PKA. Is that clear to everyone? Cool. We'll go to the next one. Nice. Okay, so we'll go over this one as well. So what we talked about, and also I want to remind you guys, like pay attention to the axes when you're looking at these graphs because they can look kind of similar, but your axes can say different things. I keep getting like distracted. Okay, so the answer is there's no more substrate left to convert to product, or like all the enzymes are busy. It wouldn't be there's no more enzyme left in the cell because the enzymes are still there. Um, the enzymes are still there, they're still present. You're not really going to see like a decrease in the enzyme presence in the cell. However, you will see a decrease in substrate presence as it's turned in the product, right? So what this graph is showing us is product concentration over time. So the reason why this graph is going to plateau is because all of the substrate has been converted to product. So once it starts plateauing, that means there's no more product left in the cell that needs, or there's no more substrate left in the cell that needs to be converted to product. Does that make sense to everyone?
These graphs are used to build the MM graphs.
Yeah.
The Kelly-Smith
Yeah.
graphs, right? This is the graph that where you take the initial velocity, just based on like that initial velocity, and then you take the initial velocity across a whole host of substrate concentrations, and then you plot the initial velocity over substrate concentration, right? And that's your Kelly-Smith equation, Kelly-Smith graph.
Yeah. So big reminder for any graph in any class, check your axes before you start thinking about what this answer could be. On to the next.
The more substrate, the more.
Okay, yes. And the answer is false because we see that it's already plateaued. So if you keep adding more and more and more, it's not necessarily going to make it go faster. We've already reached our point of saturation.
Right, so that's about more substrate, not more.
Ooh, on fire. Yes, so this one is tricky. This one was actually from her lecture. So if you want to go hear it like from Laura's mouth, it is in one of her lectures. But essentially the reason why it's like 60 and not 68 is because we're looking at the initial velocity, and the initial velocity isn't increasing anymore at 68. It's reached its maximum.
That's 60.
Huh?
You said that's 68.
Oh, okay, wait, I'm just gonna start over because now I don't remember what I said. And so it has reached its maximum initial velocity at 60. It hasn't increased anymore at 68 millimolars. I think that's what the is. So essentially we would say that it's reached saturation at 60 because the initial velocity isn't increasing anymore. Does that make sense to everyone? Okay, yeah, good job, guys. Yeah, so phosphorylation can have like any effect. It can turn something on, it can turn something off, it can just alter its function. Because it's changing the chemistry of whatever it's being added onto. It's making it more negative, like we talked about, and depending on what the function of the protein being phosphorylated is, they can have different outcomes. So it can turn something on, off, it can just change the function a little bit. Yeah, does that make sense to everyone? Cool. Okay, yeah, good job, guys. So if TK1 is always being phosphorylated, it's constantly phosphorylated, then the pathway is just gonna keep going through and through and through and through and through. Are there questions in the chat?
So is initial velocity near zero on the graph? I'm assuming that they're talking.
What was the question?
Is initial velocity near zero on the graph? We can get that. I can go.
Okay,
Yeah,
I don't remember that one off the top of my head.
well, the answer is that if the axes are substrate concentration over time, or
Mhm.
product concentration over time, then yes, initial velocity is at the beginning. If the axes are initial velocity over substrate concentration, then the entire thing is initial velocity just over substrate concentration.
Oh, I did not, I was interpreting that as like, is the initial velocity zero? I think that's why I was
they're
like-
saying years. Oh.
You could ask. Could you clarify that a little bit and then we can answer it for you? And but to go on to the next question. Question. Okay, so we'll go over this one. So the question was, when TK2 is activated, it phosphorylates and activates TFA. And I asked if this is a positive feedback loop, and the answer is no, because this is not an example of a downstream component regulating an upstream component. This is just the regular path of this cell signaling pathway. So say if it was an activated TFA, then causes the phosphorylation of TK2, because TFA is downstream and TK2 is the upstream element. If TFA was causing activation of TK2, then we would be seeing a positive feedback loop. But because what's being phosphorylated is downstream of TK2, it's just the regular pathway. Does that make sense to everyone? Is there stuff happening in the chat? Or is that you?
Then would a negative feedback loop be the opposite of positive feedback loop?
Just is negative feedback loop opposite of positive?
Yes.
Yeah. So if we were going to talk about an example of a negative feedback loop, it would be like TFA, which is TFA, say once TFA was phosphorylated, this caused something to happen where TFA would then block TK2 from being phosphorylated, therefore it couldn't be activated and it would be shutting down that component of the pathway, then that would be a negative feedback loop. Does that make sense?
Can you also, the question, yes, it does make sense. The other question was how do you find the initial velocity
What do you mean?
for like a graph? It's basically just if you have product concentration on the left and time on the right, the initial velocity is just the very first, if you draw like a linear line at that, from that like 0 point basically, that's the initial velocity at
Yeah.
that substrate concentration.
Let us know in the chat if you couldn't hear that. Scared my laptop's gonna die soon.
Oh, they did.
It couldn't.
No.
Okay, so we'll go over it after this. Or we'll just go over it at the end so we can look at the chart again. I feel like. We'll go over that question about the initial velocity at the end so that we can pull up the graph, but we have to get through questions first. Okay, I haven't even read the question. An activated TFA upregulates transcription of a kinase that phosphorylates and activates. Yes, okay. So yes, this is the positive feedback loop because we're seeing that TFA is downstream of the of RTK. So if something is happening with TFA that's causing activation and phosphorylation of RTK, this is going to be an example of our positive feedback loops. What else did people say? Someone said neither, and someone said negative feedback. So it's not negative feedback because it is promoting the pathway. It's causing us to activate the pathway, and it's not neither because it's the downstream element affecting the upstream element. So it's not just like the regular movement of the pathway. So RTK, if we pull back up the picture, we see RTK is up here. That's our like binding element. Sorry if that wasn't clear. On to the next. Okay, we'll just hope for the best. Just turn my brightness down. Okay, yeah. So for this, they're all right for this one. So the G1 checkpoint is just checking for like basic necessities, like the cell is big enough to sustain the cell cycle. It has the nutrients it needs to go through the cell cycle. The DNA is undamaged and like is good to be replicated, and also that the cell has received the signal prompting it to divide. Okay. Does that make sense to everyone? So it's all of the above. Yeah, I circled it on the diagram. I told them it's the top part. I don't know if it was clear though. We can't go back over it. Okay, yes. So the answer is all of the above. As we talked about before, it needs to be bound to cyclin to be present. Phosphorylation and dephosphorylation plays a role in activating or inhibiting or deactivating, I guess, the CDK. And then there's also the CDK inhibitors that will also regulate the function of CDK. Ignore the grammar on this one. I hit the word limit, so I had to make it like choppy. But the enzyme required for DNA synthesis is mutated such that DNA can no longer be synthesized. What checkpoint would this activate or trigger? So yes, S phase is correct. So we know that S phase is responsible for replication of the DNA. So that requires DNA synthesis. If the enzyme for this is mutated, that doesn't allow this to happen, then we're going to get stuck in the S phase, and the S phase checkpoint is going to hold the cell there. Is that clear to everyone? Cool. Nice. Okay, so the answer here is apoptosis. So we know that like P53 is one of our tumor suppressors. This is one of our breaks, right? So if we have a gain of function mutation in one of our breaks, we're not going to be going through the cell cycle at all. We're just going to be dying, basically, because it's going to be held back. So it'll undergo cell death. So this, I mean, honestly, I would write on my cheat sheet, like, what are proto-oncogenes, what are tumor suppressors, because you're really gonna want to know the difference between those two to like answer the questions accurately on the test. Because if you have those like mixed up, because I know I always get this one mixed up, because in my mind I'm like proto-oncogene P53, they have the P's, they must be the same thing, and they're not. So like, this is something I would write down personally. Does that make sense to everyone? And I don't mean like mutation, I mean like in their current state. Close, yes, good job. So the proto-oncogene is like the good version, the regular version, non-mutated, and then your oncogene is going to be your mutated cancer-causing gene. Yep, perfect. So we know that RB is another one of our tumor suppressors that stops us from going through the cell cycle. So if we have a mutation in RB, the cell's not going to be stopped basically, and it'll just keep going through the cell cycle and keep on dividing. And can this go away, please? Okay, and that was the last question. Good job. Good job again. Yay, good job. Sophia. Perfect. Okay, so that's everything. Do you want me to just pull it?
There's a picture, but if you go up, you will click question.
What if I go to this? Does this say names? Okay, whatever. I'm just gonna go here. I'm just scared my laptop's really gonna die now. This one?
No, it's too up.
This one?
Basically, if you have product concentration on your y-axis and time on your x-axis, then the first part of your graph is going to be linear essentially, and then it's going to kind of curve, and then it's going to level off as you use up all of your substrate and turn it all into product. So that linear part is the initial velocity, but this is only if you have product concentration on your y-axis and time on your x-axis. What you do is you do this, you make this graph across a whole bunch of substrate concentrations, and then so you get the initial velocity for a whole bunch of substrates, and then you graph initial velocity across substrate. So at substrate 0.5 moles, you have this initial velocity. At substrate 1 moles, you have this initial velocity. And that is how you get, where's your mouse?
It's
Yeah.
right there.
No.
It's hard to see. 'Cause I didn't
That's how
write
you
this
get
down.
this graph, where you have initial velocity on the y-axis and substrate on the x-axis. It is a little confusing because the shape of both the graphs is pretty much the same, which makes it hard to differentiate between the two sometimes, but just keep in mind what's on the y-axis really, because that'll tell you if you're looking at initial velocity there or product concentration. So,
Does anybody else have any questions about anything we talked about tonight?
In that case, we're going to stop. The graph is initial velocity of the y-axis, but that shows calendar. No, it's just analytic.
Should I stop sharing?
Yeah.
Cool.
So we're gonna stop the recording, and if you guys have any other questions, feel free to ask them. Or if you guys were